I guess I'll start by describing what my lab does, and then I'll talk a little bit about some old data and then talk about the new data. The title of my talk says that I'm going to go from rats to risk markets. I'll try to do that. There will be a lot of gaps in between. But the talk is really about choice and about risky choice. So I think whether you're an academic or a trader, you would also agree that financial events can induce emotional responses in people. I think what's more controversial and interesting is the contention that emotional responses can actually influence choice. Now, many of us have the intuition that this is the case, but it's actually difficult to demonstrate. And so one part of my argument this morning is that, in fact, emotional states can influence choice, and we can use new tools of neuroscience to demonstrate that, and even to get some causal purchase on how emotional states can influence choice. So, to provide some background, these are two questions that my lab has sought to address over the last 15 years, and as part of various nascent fields, affective neuroscience and neuroeconomics. So the first question is, which neural substrates or brain mechanisms anticipate good and bad events? And this is a central question of affective neuroscience, which we can approach with both animal models and human data. The second question is, does the activity of those circuits influence choice? And that's a central question of neuroeconomics. Now, you can think of these questions just not as conceptually sequential, but also temporally sequential. That is to say that most of the work that I'm going to refer to that addresses the first question occurred between 2000 and 2005 or 2006, and much of the work that I'll refer to that addresses the second question occurred from 2007 and on. And there's an additional set of questions that I'll get to later in the talk that is occurring now. But you can also think of these questions in terms of constructing an experimental trial. So you can think of, and also a very simple model of the brain, with apologies to Chris over there. So that input goes into the brain, it causes correlated activity. Some of that activity is predictive of the output, okay? And what's interesting from an experimental standpoint is, can we identify these overlapping, this overlapping area in these circles? Can we identify not only the activity that is correlated with the input, but it actually predicts the output? So you can set up an experimental trial with the arrow of time going from left to right, and if you can visualize activity associated with both of these events, then you can have a chance at sort of identifying the mediating neural substrates. At least that's the logic that we've been using. Okay, so I want to first describe some work that we've done in individuals trying to use brain activity to identify these correlates that are responsive to anticipating good and bad events, often using money, and then that predict choice. And then I'll try to describe some a study at least, that we've done in collaboration with Carl Dijsselrath's lab, where we actually try to control individual choice by causally intervening in the substrates that we've identified in rats instead of in people. And finally, I'll describe some more recent studies where we try to link up to describe neural predictors not only of individual choice, but also of aggregate choice, and in this case, stock market price movements. Okay, so but I have to start with this slide. So this is a Canadian slide, of course. This picture was taken at McGill University. It depicts a rat that was owned by Donald Hebb's graduate student James Olds and Peter Milner. The rat has a wire in its head. It's pressing a bar. It will continue pressing that bar until it becomes exhausted. It will ignore other rats. It will ignore food. It will ignore drink. It will ignore sex. It will ignore sleep as long as it can, and will just press that bar. This is a phenomenon called self-stimulation that was accidentally discovered in Hebb's lab, but turned out to be quite interesting from a motivational theory perspective. And so when Olds found this, he was very excited, and he said, I found the reward circuit. And there was something to that, but actually the story gets a little bit more complex, as we found out in later years. So how does it get more complex? Well, for one thing, the substrates that elicit this self-stimulation behavior, not only in rats, but in monkeys and even in people, can be mapped electrically or with chemistry. And they tend to lie along the ascending trajectory of midbrain dopamine neurons that emanate from the ventral tegmental area, it's a little rendering, up into subcortical areas like the striatum, and also into prefrontal cortical areas, like the medial prefrontal cortex and the orbital frontal cortex. And this is an interesting fact, and it implicates the chemical dopamine in this behavior. Of course, there is a complicated chemistry and neural circuitry that combines to make up this circuit. Another slide, which is the most famous slide in neuroscience, I think, and I'm contractually obligated to show you, is the Wolfram-Schultz slide, which shows that if you record from these dopamine neurons, when a monkey gets a reward, like a squirt of juice, then indeed they will fire. So this the raster plot showing the firing of the midbrain dopamine neurons, monkeys getting a squirt of juice, and they increase in their firing. So that looks like a reward neuron, right? But not so fast, because if you cue the monkey that it may get this juice, then in fact the firing moves from the delivery of the juice to the cue. So if the monkey sees a flash of light or hears a sound, then the firing of those neurons will actually occur when the cue is presented, not when the reward is presented. So these might be like surprise neurons, right? This is something that the monkey didn't expect, and they're chasing the most surprising stimulus. But that's not the case either, because if you play a trick on the monkey and you present the cue, but then you omit the reward. So you present the cue, monkey waits, thinks it's going to get a squirt of juice, no squirt of juice occurs. There's actually a decrease in the firing of these neurons, suggesting that these neurons do code something about value. But it's not so much getting the reward as anticipating or predicting the reward, which has been called or linked to the reward prediction error idea in behaviorism. Even though this is an amazing finding and very interesting, it doesn't tell us where the dopamine release is mattering to motivate behavior. So these dopamine neurons, as I mentioned, project up into subcortical and cortical regions. So these kinds of studies can tell us what's happening at the dopamine neuron cell body, but they can't tell us where the action is then translating into approach behavior, for instance, or tongue licking in the case of the monkey. Fortunately, in 1992, methods were developed that sort of hit a sweet spot of temporal and spatial resolution that allowed us to look into the brain, deep into the brain, below the cortex, at a time scale of seconds and at a spatial scale of millimeters. I'm, of course, referring to functional magnetic resonance imaging, which most of you are probably familiar with here. And it has slightly better temporal resolution than positron emission tomography and other methods that were available at the time. Slightly worse temporal resolution than electroencephalography, but again, you can go deep and get this good spatial resolution and decent temporal resolution. Good enough temporal resolution that you can start to ask the question, not only what happens in the brain when the monkey gets the reward, or the person in this case, but also what happens in the brain before the person gets the reward or the monkey gets the reward. And so when I was on my third postdoc at the National Institute of Health, my advisor said, hey, we have some time on this fMRI, and do you want to use it for something? And I sort of jumped at that opportunity. And we wanted to try to visualize where these dopamine neurons were projecting, those areas, and the activity of those areas. Of course, we're looking at oxygen, not dopamine, with BOLD. And we were faced with the challenge of what kind of stimulus can we use that will motivate people reliably and that they won't sate on. And of course, there are huge individual differences in what motivates people. Some people like tofu, others like steak. So it's very difficult to select a standardized set of pictures or even tastes that will continuously motivate people. And we hit upon the idea of using money to motivate people. And this seems to motivate almost everybody, even grad students, and in Palo Alto and also in Washington, D.C. And so this was actually a very handy experimental tool for us using the neuroimaging. And so we were able to set up a probably the dumbest video game ever, where we could show people a cue that indicated they might be able to make money or avoid losing money, and we could indicate that by cue features like the shape of the cue. People wait a couple of seconds, and then they see a rapidly presented target, boop, on the screen, and their job is simple. It's on every trial, they just have to press the button before the target goes away. If they're successful, after anticipating gain, they will make money, and if not, they don't. If they're successful after anticipating loss, then they will avoid losing money, and if not, they lose money. So this allows us to look, because of the temporal staging of the paradigm, not only at what happens in the brain when people get the reward they expected or don't, but also what happens before they ever get the reward. And how do we control that? Well, we can actually control the timing of the target so we can adapt it to the user's performance so that they're going to succeed more often than not, but say 66% of the time, but not always, so they stay engaged in the video game. And of course, it's for real money, so this was hard to get by the Human Subjects Committee, but I actually paid people in cash for their performance afterwards. And so we continue to do that to this day. So as I mentioned, you can manipulate many aspects of the trial design, not only whether people anticipate gaining or losing, but also how much they might think they could gain or lose with a 66% probability. We can also manipulate other aspects of this too. And it turns out this is of interest to economists. I didn't know it at the time, but there's a construct called expected value or expected utility that could well be related to this. And it turns out it's easy to manipulate these Q features to approximate features of expected value. Okay, so to cut a long story short, we did a lot of research on this, and others now have done a lot of research on it as well. And this is kind of what we found. So we found that activity in certain areas of the brain increases before people think they're going to get the reward, or before they get the reward, I should say, when they think they might get it. And those areas include this area right here, the ventral striatum, a part of it called the nucleus accumbens. Okay, and so the more money you think you can make, the more activity we see in that region, right before people respond to get the reward. There are other regions, like this anterior insular region, where we see a more symmetric response. That is, the activity goes up as a function of the magnitude of anticipated gain or anticipated loss. Now, what's interesting about this orange region, the nucleus accumbens, is that it's one of the projection sites of the ventral tegmental area, which is one of the midbrain dopamine nuclei. And we know from animal studies that it's very important, dopamine release in that area is very important for motivated behavior. So we're able to actually capture a signal that we think is related to this, and now we know it is, but I won't go into that. We think is related to dopamine release and could be related to motivated behavior. Of course, I haven't shown you that yet. We're also able to parametrically activate that area with these financial reward cues. So this might then have an implication not only for anticipation, but for what you're about to do next, okay, in the following way. If you anticipate more gains, so if we think about this in the context of risky choices involve something potentially good or you wouldn't be interested, but they also involve something potentially bad or it wouldn't be risky. Okay, so you sort of have to balance those anticipated gains and losses. So what if we could look in the brain and see how your brain is balancing those anticipated gains and losses, those uncertain gains and losses? That's what we think we could do, because if this area is responding to anticipated gain and that to anticipated loss, then reversing the traditional logic of brain imaging, if you look in the brain before people make a choice and use that activation, you should be able to predict the next choice. So if it's a risky choice and there's more activity in this orange area, people should be more likely to approach the risk. And if there's more activity in this blue area, perhaps they might be more likely to avoid the risk. There's another wrinkle in this story that involves the prefrontal cortex, which we think is playing a role in balancing out these competing drives. So these are really, we think, markers for predicting behavior. Do they predict behavior? This is what I just described. Yes, they do in many different scenarios. And Britt talked about shopping. We've actually looked at shopping. We've also looked at risky financial choice scenarios, including gambling and investment. I'll show you a gambling example. But first, I'd like to probe your intuition on this. So here's a gamble right here that I might offer you. How many people would be willing to take this gamble? 1, yes, we have a risk taker. 2, okay, we have at least 2 people. Okay, good. This shows there's diversity in risk preferences. Okay, how about this gamble? Who would be willing to take this gamble? Slightly more, but not that many. I'm seeing like about, I don't know, 20 hands going up, scattered about the audience. Okay. All right, how about this gamble? Who would be willing to take this? Many more hands are going up. I would say almost 50% of the hands are going up. So, and this is the common pattern that we see in experimental subjects. Now, an interesting thing about this gamble from a financial perspective is that they are equivalent in terms of their mean and in terms of their variance. So that means most financial theories that are used to construct your portfolio will treat these gambles as equal. And yet, most of our subjects do prefer this gamble, which I'll call the positive skewed gamble, right up here, to this gamble, which I'll call the negative skewed gamble, which is interesting, and in some frameworks, suboptimal. And yet, we don't feel bad if you show this pattern, because most people do. So why is that? Can this neural data tell us something not only about why people make risky choices, but also when they deviate from rationality or optimality? Yes, the answer is yes. So we did this experiment with fMRI. We showed people these different gambles. repeatedly. We asked them if they wanted to accept them or not. They played for real money. They were shown their total. And we looked at brain activity during this anticipatory period. So you can see that we're always sort of pairing a gamble with just nothing happening. And people can either choose to take the risk, yes, or choose not to take the risk, no. What we want to predict is when do people choose to take a risk. And this is actually a classifier. So this is not a theory-driven analysis. But if we do a theory-driven analysis using those volumes of interest, we get the same result. When you throw a classifier at the data and you say, classify the features in the brain or the voxels with activation that separate out whether you're about to take a risk or not, this is the map we get. So what you see is it's pulling out features in the ventral striatum that are predicting you're about to take a risk. It's also pulling out features in the anterior insula and parts of the anterior cingulate, or maybe further up, they're predicting you're about to not take a risk, okay? So it's not just all the brain is predicting you're going to take a risk and it's silent if you're not. There are some features that are predicting yes, some features are predicting no here. And you get highly significant results with using this classifier that generalize across subjects, basically. So this generalizes to different people. More interestingly, though, what about this weird pattern that you guys showed where more people preferred the positive skew than the negative skew gamble? Well, we can also account for that with the brain data. So if we extract brain data from those volumes of interest in the nucleus accumbens and the anterior insula, what we can show is that these are some other features of the experiment that might matter, like how long have they been doing it, how much have they earned, what's the previous outcome. But we control for those, and we look at the gamble type, and we add the brain activity. The brain activity actually eats up the variance. So there's a main effect of nucleus accumbens activation and anterior insula activation, as I showed you. But there's also an interaction between nucleus accumbens activation and the gamble type. And what that indicates is that overall, in trials where the positive skew gamble is in play, the nucleus accumbens activation is greater, and it's predicting greater choice of those gambles. So this is just an example of how we can use brain activity to predict individual choice. And there are many other examples, but I think I'm using this one because it's going to tie into a later experiment that I'm going to discuss. You might question my experiments, and I wouldn't blame you if you did. But many experiments have been done. Actually, Olaf Sporns and I were speaking at the coffee break about there are actually replicable results in brain imaging, thankfully, since it costs a lot of money to do. So you can do these meta-analyses using various databases like Neuroscience and ask, if you were to look at this location in the brain, which terms would pop up in abstracts of studies that describe the activation there, and the reverse way too. And so this is actually an ALE an activation likelihood estimate meta-analysis that we did of studies on anticipating financial variables. So enough studies have been done that we could do this where the mean was manipulated of the risk or the variance was manipulated of the risk. And you see this pretty decent separation in this ALE meta-analysis. But you can also go to databases like Neuroscience and just type in the word reward and look for what kinds of brain activation are associated with that. And you see a similar kind of a map here of the nucleus accumbens. This is 671 studies that are in the Neuroscience database. And you can also type in words like pain, and you see a kind of a different matrix popping up here, more related to anterior cingulate, anterior insula, and anterior cingulate activation. That's 420 studies. The Neuroscience database also has a neat feature where you can actually look at a functional connectivity in a data set of 1,000 that was provided by Randy Buckner's lab. And so I just was fooling around with it, and I thought, what happens if you actually just land your cursor right in the nucleus accumbens? What's positively correlated, but more importantly, negatively correlated with that, because this is often not discussed in the functional connectivity literature. And what you see is this really surprising and clear pattern of negative connectivity with the anterior insula, which I hadn't been aware of, actually. And conversely, if you put your seed in the anterior insula, you see negative connectivity in the nucleus accumbens. So I found this quite interesting, too. It's not something we've looked at, but since Olaf is here, I think it's worth looking into, and I think it's worth taking these negative connectivities as seriously as we take the positive connectivities, even though we don't have a great physiological model for what's going on. So having said that, I hope I've convinced you that we can use brain activity to predict individual choice, individual financial risky choice. But this has led to other questions and other directions of research. And so I characterize these directions of research as going down a level and going up a level. So going down a level, we can ask more specific physiological questions, even causal questions, like does perturbing the activity in some of these circuits actually influence risky choice? And does it do so on a trial-to-trial basis? We can also go up a level, and this is the level that my economist friends are interested in, and ask what is the relation, given that we can use brain activity to predict individual choice, can we also use it to predict something about aggregate choice, even market choice? And this is a little bit kooky, but that's the new data, but I'll show you some data that I think is interesting in that respect. Okay, so I'll start with linking down. So what we're trying to do here is to control risky choice. We have some targets in the brain. Ideally, we would go in and just dump dopamine in the nucleus accumbens. That would be invasive and hard to get by human subjects. However, there are also rat models and rodent models of risky choice that we can actually borrow from the human models, right? And there are also these new techniques like optogenetics and fiber photometry, which will allow you to perturb and observe specific cell type activity in rodent models. So we were very lucky to be down the street from Karl Dijsselrath, who is also a great colleague and a good collaborator, and really Kelly Zalakuski, who is his grad student now as a postdoc, was the one who made this happen. So all the credit belongs to her, with her. But she was very convinced that we should be looking at risky choice in rodents and convinced Carl to do it. And I convinced her to use rats because rats can deliberate. And mice have a harder time deliberating. And we needed the rats to deliberate about these risky choices. So I showed you that risky choice paradigm in humans, right? We would basically use the same paradigm in these rats with the same timing. The only difference is we didn't use money, obviously. We used juice. So we were able to present rats with the same kinds of gambles. You choose one lever, and you always get 50 microliters of a tasty fluid. Or you choose the other lever, and you have a small chance of getting a lot of fluid and a large chance of getting not much fluid at all. All right, and the rat just makes these choices sequentially. And the rats do sort of allocate their choice. They don't always choose the same thing. They sort of switch around, but they do have preferences, as you'll see in a second. So some rats tend to like, as you guys also show diversity in preferences, some rats tend to like the low-risk option. Other rats tend to like the high-risk option, a smaller group. Yes?
With the money, the negative money.
Yes.
Less
Yes.
here it's just less. Oh, is that the same?
It's less than the expected value, and it's not exactly the same. However, we're trying to manipulate the nucleus accumbens in this experiment, so it's one of the beautiful aspects of money is that you can easily present losses and gains in the same modality. With animals, it's harder. So we haven't quite worked out how to trade them off in rats, but it's a noble undertaking, and we're trying it. So yeah. Yeah, this would just be sort of not as good as you expected. Yeah, so I would call it non-gain. But yeah, it's an excellent point. But at any rate, they do sort of act like humans. They sort of allocate their risk. They change their choices. Some like risk, some don't. You can also see that in the rats that are risk averse, they're more affected by getting less than they expected on the previous trial. Okay, so and you can model this with reinforcement learning algorithms and show there's not much influence from the past trials, but usually it's the last trial, what happened on that trial, that tends to influence the rat's next choice. Okay, so here you see a distribution of the animals that were risk seekers and risk avoiders, and here you see that the risk averse rats were less likely to take a risk on the next trial if they had not gained on the previous trial. They had taken a risk and not gained. Okay. You see that the risk-seeking rats are actually kind of impervious to this. So they don't tend to weigh relative losses on the last trial as much. So can we see what's happening in the nucleus accumbens as this is occurring? And the answer is yes, but only in a limited way. For actually around the time we did this experiment, Carl's lab developed a technique called fiber photometry. And this is a technique which is like magic to me, but in it you can transfect specific neurons that express, say, D2 receptors and cause them to light up when they fire. And then you can stick in an optical probe, and with appropriate controls, can observe changes in the firing of that population of neurons in the specific brain area, like the nucleus accumbens. Now, the D2 neurons are kind of interesting because they tend to inhibit postsynaptic activity, but they use energy. Okay, so D1 expressing neurons tend to get excited when dopamine hits them, and D2 expressing neurons tend to get inhibited when dopamine hits them. And there's a whole theory that some of you may be familiar with about the relative roles of these neurons. This is the construct they had, though, one that could hit the D2 neurons in the nucleus accumbens. So this just shows the apparatus and the cool techniques they developed for ruling out motion confounds by using two separate bands of light. But they were able to actually visualize the firing of the D2 expressing neurons during this paradigm. And what they found was that after rats had taken a risk and experienced a relative loss, there was more firing of these D2 expressing neurons. And that this was particularly the case for the risk-averse rats. This suggested that if we could go in and we could intervene on those neurons, we might be able to push around risk-seeking choices. So that's what they did with optogenetics. So they had a construct now that they could transfect these D2 expressing neurons with that would make them responsive to light. So then you can put in an optical probe that will shine at a specific wavelength, which will activate these neurons, these D2 expressing neurons, right before choice. And the question was, could they actually perturb choice? And the answer was yes in the risk-seeking rats. So in the risk-averse rats, they're basically at the floor. So if you perturb these D2 expressing neurons, they're still not going to take much risk. This is just the control group. So they have a control construct that's expressed in the nucleus accumbens. But this risk-seeking group, you can see they're pretty high off the floor, and choosing the risky option on the day when you're stimulating these D2 expressing nucleus accumbens neurons, they take less risk. They bounce up a bit on the next day, and the next day they bounce down again when you're stimulating the D2 expressing neurons, and then they bounce up again. This is not the case with the control construct, which does not make the neurons responsive to light. What's most interesting from my standpoint is that you can observe this on a trial-to-trial basis. So of course they didn't stimulate the rat on every trial, they did on some of the trials. And so you can compare sort of what happened before and after they stimulated. And this is interesting because there are different accounts of what's going on in the nucleus accumbens. Is it that we're changing preferences, as the economists would say, or are we changing beliefs? Like, are the rats learning something? You know, that should be, if they're learning something, there should be a lasting effect. If we're flipping preferences, then it might be able to flip on and off at a faster rate. What we see is the latter. So here are the trials before rats receive the stimulation. You see, again, these risk-preferring rats are responding at a fairly high normalized rate. And then you apply the stimulation on that trial, they are less likely to choose the risky option. And on the next trial, if anything, it bounces up. So this is sort of inconsistent with a long-term learning perspective. This suggests that there's a trial-specific manipulation going on here. Yeah?
Manipulation only on before the rats get feedback or also during feedback?
Good question. Yeah, we did both, but we found the most powerful effects right before the rats chose. So I didn't probably describe the paradigm well enough, but it's sort of like the fMRI paradigm. So the rats were trained to know on that day which was a risky lever and which was not. And then they were presented with both, but they had to wait, and then they could nose poke for one or the other. So when they were waiting and contemplating their choice, that's when we applied this stimulation. Great question. Yeah. Okay, so this is kind of science fiction. There's much more you could do with this, and there is more that we have done actually, trying to link actually optogenetic control of dopamine neurons to the bold signal, which I could talk about later if you're interested. But now I'm going to flip to something up a level, 2 levels, and ask the question, oh, maybe I should summarize this. So rats vary in risk preferences. NACD2 activity from fibrophotometry increases prior to risk avoidance. Stimulating those neurons prior to choice reduces pre-existing risk seeking in these rats. What I would love is to be able to control the NAC, the nucleus accumbens D1 expressing neurons. These are the ones that, in theory, should be driving the risk seeking. We don't have the tools yet, but knowing that lab, they will soon. Okay, but let's now talk about 2 levels up. Can we forecast something not only about individual choice, but also about aggregate choice? So we've been playing with various forms of this data, and we realized that there's a dearth of explicit theories about how individual choice should link to aggregate choice. Or maybe I'm just ignorant of those theories, and if I am, I want you to tell me afterwards so that I can read these theories. But in a stylized continuum, there are two extreme examples, right? So one example is basically individual choice will tell you nothing about aggregate choice. And why might that be? Well, people might have consistent preferences, even biased preferences, but if you look at everybody together, they form a normal distribution, and everything comes out in the wash. Either that, or there's game theoretic in the stock market, for instance. If I have a bias, somebody else will recognize that and take advantage of it, and everything comes out in the wash. So two different explanations, but the same outcome, that individual choice doesn't tell you a whole lot about aggregate choice. And so this is exemplified, for instance, by the efficient market hypothesis. So this is not, it's not a crazy theory in some ways. Another possibility is what we call total scaling. So that is just like, if you want to know what the market's doing, just measure what I'm doing and multiply, right? And this, for example, is endorsed by accounts like expected value or expected utility, implicitly. So neither of these seems very plausible, actually. So it might be that something in between is actually more plausible. And one account we've been playing with is this so-called partial scaling account. So this idea is that if you could deconstruct choice into components, measure those components, some of those components may generalize to aggregate choice better than others. So this is a little strange. It has some paradoxical implications. But it also suggests that it might pay off to actually measure brain activity as well as people's choice in a group when trying to predict aggregate choice, or forecast aggregate choice, actually, because it's out of sample. So I'm going to describe some new data from a stock pricing task that was done by then a postdoctoral fellow, Marisa, who's now a faculty at the University of Leiden. And in this task, what we did is we took 14 real stock tickers with 10 updates each, preceded by a 20-tick history. There's no other information about the stocks. And we presented these to subjects in the magnet, and we presented them with each update, and on each update, they basically had to take a risk or not. They had to basically tell us whether they thought the stock was going to go up or not. If they thought the stock was going to go up, they could take a risk, and if they were right, then they would invest $1 and they would make $2, and if not, they'd lose their $1. Or they could not take a risk and sit that trial out. So it's a very simple risk-taking task. The efficient market hypothesis makes a clear prediction about what should happen here. Basically, people should choose randomly, and their choices should not tell us anything about what the market price will do next. Okay. But is that really true? So if you were in my magnet, you might see a ticker like this. And then you might be asked to make a choice. You might think it's going to go up. You would say yes. And it did go up, so you just made $1, and your balance went up. And now you see the ticker again, and it's updated. You see the little tick went up. And so you have to, again, make a choice. You might say yes again. You're feeling risk prone, and you find that you made some more money. Okay, so that's how the task works, basically. And people know these are stock tickers. They don't know which stocks these are. So we can ask a question. The first question we can ask is, like, given people's performance, their choices, does that tell us anything about the movement of the market? And the answer is, maybe it's a relief to the efficient market hypothesis theorists in the audience, no. So in experiment 1, we found this. We did another experiment. I'll explain why in a second. And in experiment 2, no. So people look like they're choosing it about chance. Okay. But remember, we're scanning people while they're doing this, so we can actually harvest their brain data as well. Okay. So, well, maybe people are just giving up, right? They're in this experiment. It's hard. Their money is at stake. Maybe they're just like, forget about it. I'm just going to choose randomly, or they're not responding to the incentives, right? So we can look in their brain and say, okay, when they made money or they've lost money, they took a risky choice, do we see activation in the substrates I just talked about? The answer is yes, quite robustly. So when they make versus lost money, in both experiments, we see quite robust activity in the nucleus accumbens and other parts of the striatum and some other areas too. So they seem to be engaged in the task. This is also true for counterfactual outcomes. So these are not the things that really happened, but the deflection between what you expected to happen and what actually happened. So for instance, if you thought you were going to make money or you took a risk and you didn't make money, then the activation actually goes down instead of staying at baseline. So anyways, you see the same pattern of activation for these counterfactual outcomes. Okay, so people are engaged in the task, at least their brain is. They seem to be responding. Is anything else going on? Well, what if we take brain activity as I was describing and try to predict each person's choice. Will they take a risk or not? We can do that too. It's pretty weak in the first experiment, but it's in the region where we'd think. And it's a little bit stronger in the second experiment. So they seem engaged in the task. Can we use the brain activity to forecast the next price movement? Okay, this is just showing what I just described, so I won't go into it. And sorry about these, these logistic regressions. So this is, let me describe what this is. This is trying to predict not their next choice, but the next ticker movement, the next stock price movement, okay, based using a number of variables. In experiment 1, these are the market variables, so this is sort of the slope of the ticker. This is the volatility of the ticker, how much is it jiggling around. This is what happened on the last trial to them. This is the choice that they made on the last trial. This is an activity out of these volumes of interest that we're interested in. So this is the nucleus accumbens, the anterior insula, and the medial prefrontal cortex. And this is everything together plus an interaction term for the activity and what happened on the last trial. And in the first experiment, what we see is that actually, yes, it looks like nucleus accumbens activity is predicting direction. That is, if there's more activity, the price is going to go up, and if there's less, it's going to go down. Anterior insula activity is doing something more interesting. It's interacting with what happened on the last trial to predict what we, forecast what we call inflection. So if anterior insula activity is high and the price is going up, it's more likely to go down. If it's going down, it's more likely to go up. But there's a problem. In this data set, we actually have some autocorrelation. So that is that there's some information in the price that's going up and down. And people might be able to take, or the brain might be able to take advantage of that. So we conducted a second experiment with a different set of stock tickers from a different month of a different year, and that did not exhibit this autocorrelation, and reran the experiment. This is just showing what I just described, but in a time course form. So, and again, this is sort of showing that when people are looking at the ticker, nucleus accumbens activity is higher if the price is about to go up. And it flips actually for the anterior insula. So that is to say, if the price is going up and it's about to go down, it's higher. If the price is going down and it's about to go up, it's higher. So here's experiment 2. This is the logistic regression for that. And what you see is, yes, indeed, we did get rid of the autocorrelation. But unfortunately, our nucleus accumbens forecast is also gone, which I think is interesting, for a reason I'll say in a minute. However, the anterior insula term is still there, and it's still sort of doing the same thing. So even in this case, the anterior insula activation seems to be telling us something is about to change in the ticker. Yeah? Yeah, it's pretty small. Yeah, that's right. No, so we have done, this is significantly different than that, for instance, but it's still not a very big fit. So, we're not actually getting much out of anything except for this one term. So it's not that impressive. However, I think with stock market prediction, you have to ask compared to what. So the efficient market hypothesis makes a clear prediction that chance is the baseline, right? So even though it may not be very impressive, it's interesting to us that anything might forecast stock price movement. And this is probably not a very well optimized experiment because it's the first time we've done this sort of experiment. So our parameter is there are many levels of variance, so some of which I'm ignoring here. And so we may be able to get more out of our signal. In fact, I think it's a general theme, maybe that would be controversial to some, but that people aren't getting all of the signal that they can out of their fMRI data because they're sort of collapsing over time in various ways or fitting to the data in ways that sort of omit useful parts of the data. Because these signals are flipping around quite fast on a second-to-second basis, and not in a random way, in a systematic way in these paradigms. So I think there's more. So for instance, I showed you the classifier models, right? That's a theory-free way of extracting features in both space and time that can allow you to test your theory. Like, am I choosing the right volumes of interest at the right time points, and to refine those theories, or just rule them out. So just to summarize this very novel data, group choice does not seem to forecast stock price in our hands, so the efficient market hypothesis wins there. However, in markets with structure, the nucleus accumbens activity may forecast price direction. Of course, we would like to replicate that. while anterior insula activity seems to forecast price inflection. And even in markets without structure, the anterior insula activity still seems to forecast price inflection. So when I say forecast, I don't mean predict that individuals, I mean forecast to the aggregate level. So what have I tried to convince you of? We can predict risky choice in individuals using fMRI in humans, and this is new as of 15 years ago. We couldn't do that before we had the technology. We also, I think, are beginning to control risky choice on a single trial basis in rodent models with specific cell manipulations. And this is really exciting to me because it highlights the utility of using these translational models that you can use in different species and taking advantage of the technological advances that you have in each. And finally, we may be able to forecast market choice and other aggregate choice variables using fMRI in humans. And it's an open question, when is the brain signal more useful and when is the behavioral signal more useful? I spoke in psychology about this yesterday. We don't really have a handle on that yet. So I think there are many applications to this that we haven't explored. One is like, how much control do we have over these neural substrates? These are fairly deep neural substrates. They're fairly conserved over the phylogenetic spectrum. So how much control can we get over them? And how can we increase people's control or awareness of what's going on, right? And that probably involves not only thinking about situations and environments, but also thinking about timing, because as I mentioned, that these signals are fairly time-specific. Can we develop, with that kind of an understanding, can we develop choice architecture, prostheses, that will help us to make better choices? They don't have to be just bionics. They could be other kinds of tools that we have readily available in our environment. And finally, this neuroforecasting idea is an interesting one from an applied as well as a theoretical sense. And there are many limits that we don't understand and potentialities there that remain to be discovered. But I think this has theoretical implications too, this kind of work. I think it moves us beyond description and explanation, which was the modus operandi in brain imaging research, to prediction and control. And those are not the same thing. Those are not the same stages of science. We need new tools for prediction and control. And we're starting to get them. We're very excited about improving the models that we have with neural constraints at all levels of analysis, and especially about linking levels of analysis. So to sort of exemplify that, this is the kind of very basic framework we're working with. What if we could say not only that we can measure neurochemical activities in the brain, but we can use that then to predict what kind of bold response a person will have that we can measure with fMRI. And we can use that to predict both what kinds of feeling states they're likely to be in, say a positive aroused state, and also the probability with which they'll approach something. And we can even use that, with some limits, to predict aggregate choice. That would be pretty neat. So this is the most important slide where I thank the people who actually did most of the work. My collaborators, Marisa, who I mentioned, Nick Borg and Charlene Wu were in the lab working on these projects as well, and Kelly Zalakuski from the Dijsselrath lab, who really made that happen. And also our funding sources, which are somewhat unconventional. We got some seed grants to do this translational rat-to-human work. And we also now have money from the NeuroChoice Initiative that allows us to ask kind of crazy cross-level questions. And thanks for being here so early on a snowy morning. I shouldn't pretend to understand finance either, in fact. But I do collaborate with some economists and some people in finance now, so who are interested in these applications and ideas. Yeah, I don't think I can answer your question, but I think I can point out an interesting feature of our data, which is if the brain is allowing us, if brain activity is allowing us to forecast something about a price movement, then something must have gotten in there, right, to cause that brain area to forecast that. Now, if the anterior insula is our most robust result in this case, what is it doing? You know, we don't really know. Now, there's a lot of data to suggest, just in pure financial choice at the individual level, that the anterior insula is responding somehow to variance. And that could be related to a lot of things like arousal, uncertainty, you know, those sorts of things. But we don't have a great handle functionally, as good of a handle, I should say, on what the anterior insula is doing functionally or psychologically, as we do, I think, on the nucleus accumbens, or as we're getting the picture on the nucleus accumbens. So there's a very open set of questions there. Also, you can ask the same question at a physiological level. You can say, okay, so what I didn't tell you is we can stick those rats in the magnet awake, and we can actually control their dopamine neurons and ask, what is the bold signature of stimulating the dopamine neurons? And sure enough, it looks a lot like that reward cue signature that you saw, or that about to take a risk signature. So the striatum increases in activation, and it's very robust. Not so with the insula. So then the question is, what's the neurochemical modulator that's driving the insula? And we think, you know, if you put a gun to my head, I would say it's probably norepinephrine, which we know responds to novelty and uncertainty in the environment in animal models. So I don't think this is answering your question. I'm sort of evading it by leaping levels down. But I do think that, like, by doing that, that could give us insight on what are the features of the price movement that are actually activating this in a way that tells us something about the next movement. Absolutely. Yeah, I mean, I'm not an expert on the efficient market hypothesis, but it seems to me to be time-free, right? And it's sort of like, well, whenever equilibrium occurs, that's the price. But it's like, when does equilibrium actually occur? Are we in equilibrium now, or, you know, will it take 40 days? Or so, but to be fair, I mean, we're sort of compressing timescales. So each of those ticks is the close of a day of trading. Okay. So, but we're presenting it on a second-to-second basis. So that's a bit of a stretch as well. Yeah. Oh, okay. Yeah. Well, I think one has to distinguish between individual prediction and aggregate forecasting in this case. So if you look at individual prediction in that what they're about to choose in the stock task, in both of those cases, we do see, like if we do a volume of interest analysis, the nucleus accumbens activity does predict that people are about to take a risk. So I'm sorry, I probably glossed over that. But the aggregate results are different, and that's interesting. That's why I think it's actually worth trying to both predict individual choice and forecast aggregate choice, because one thing that we're finding in data that I didn't describe is that it's not always the same neural substrates that are predicting individual choice and forecasting aggregate choice. In fact, in our hands and in our paradigms, it seems like the subcortical activation is more forecasting more aggregate choice, but the cortical types of activation aren't as much. So, which is interesting, but kind of a different point, I guess. Yeah. That's essentially what we think. So that, and in addition to that, so we think that there's sort of implicit detection of patterns, and that's maybe why we saw the aggregate forecasting on the first study, because there was this autocorrelation. So there was some pattern that the nucleus accumbens could detect and try to ride. That's post hoc, of course. And that's implicit. You don't really need sophisticated language or anything like that to detect and follow these patterns. But we also think there's a motivational element there, that it's not just that you're detecting the pattern, but you're actually compelled to follow the pattern. And that's why, you know, we're eliciting these subcortical activations. With respect to the cortical activation, in many scenarios, we see MPFC activation, the activation of the prefrontal cortex, also predicts individual choice, just like the nucleus accumbens does, not just like, but similar. But when we then try to make an aggregate forecast, that falls out. Now, why might that be? Well, it might be exactly what you're saying, that, you know, everyone's detecting sort of a certain pattern motivated by that, but then individual considerations come in, you know, and those are idiosyncratic, and they may make you a more consistent chooser within yourself, but they may make you less consistent with everyone else. And so that's one possibility that could help explain these partial scaling phenomena. Yeah. Yeah.
Don't take any risks, and the ones that do, are they, I mean, all like the same age?
Yeah.
Adolescent rats in there?
We haven't looked at age. I think they're mostly adolescent, early adult, and they're pretty genetically similar. In fact, the ones where we optogenetically manipulated their neurons, those are special mutants that have their neurons responsive to sort of light stimulation after a viral transfection. So yes, but it's getting to the point where we don't need mutants anymore to do this. You can actually just put in the virus and it'll find the right neurons and transfect them. Oh, in rats? It's a great idea, no, but we would love to do that. Part of the problem is the rat insula is not well mapped. So we have to actually sort of identify it and figure out, you know, which neurons in there to stimulate, but I think that's completely tractable in theory. We just need the right tools to do it. And I suspect they won't be the same kinds of neurons we're looking at in the nucleus accumbens. So yeah, it's a great idea. And that's the kind of experiment that these people who use optogenetics are starting to do on a regular basis, a semi-regular basis, yeah. There was a question over here, yeah. We haven't looked at Gambler's fallacy, but it should be embedded in our data, actually. So that's a great idea. We should look at that. What we have looked at, actually, is individual differences in anticipatory anterior insula activity and willingness to take a risk in a stock picking scenario, a different scenario than the one I described. One which is more controlled experimentally, and which we can determine what's the optimal choice given a Bayesian updating risk-neutral rational actor on each trial given the information they've gotten. And what we do find is that indeed people with more sort of anticipatory right anterior insula activity seem to overall avoid taking risks more often in that scenario. But I haven't looked at the Gambler's fallacy. That's a great idea. Yeah, so it's an interesting question because, again, you could characterize this as a very random process. People, if they were rational, might say this is a random process. I've been tracking the outcomes. Why trial? Just randomly choose the buttons. They seem engaged. Their brain looks like they're engaged. It's probably true with rats. There's an interesting question about engagement and maybe a sense of control. Like if you force choices upon the rats or the people as opposed to allowing them to choose, how does that change the phenomenon? And we haven't looked at that. That's really interesting. We actually did a passive gambling experiment a while ago, but people were less engaged in that, as you might imagine. But we didn't systematically, vary that as you might want to do if you were trying to operationalize some sense of control. And even though it sounds kind of crazy to think that rats have a sense of control, I don't think it's so crazy because humans can distinguish between when they think they're making a choice and something is just happening to them. And so I think rats could probably do the same thing. I haven't thought deeply about it.
Mine can be somewhat suspicious.
Really? Okay.
About the root of the action.
Uh-huh.
They create their own strategies
Oh, yeah.
to get rewarded.
Right, right. Yeah, and I haven't thought about that either, although I feel like B.F. Skinner wrote about it. But he probably didn't write about a feeling of control. So yeah, I'm sorry, I haven't thought deeply about it, but I can imagine tractable ways to try to get at it experimentally. But we haven't done them. We haven't done that.
Previous studies?
I'm sorry?
Are there any previous studies?
I don't know. So yeah, I apologize. Yeah.
Okay, well, let's, so yeah, we can all applaud one more time. Great.
Thank you.